Wayne's on the mic, about to wrap up a storm talking about nitrogen and how it changes form. Gonna get some things right, so let me your ear. Nitrogen is 78% of the atmosphere. It's used to take nucleic acid in all living things, from a plant to a fish to a bird flapping its wings. Nitrogen from the soil is taken up by primary producers and used by consumers. The cycle comes to a closure as it's put back into the soil by a decomposer.
Be Patty P. Daddy Blade in the house. Gonna start this rap, not gonna delay. It's about time y'all learn about your DNA. It's made out of nucleotides joined by covalent bonds. Yep, they can help make those sexy blondes. Let's talk about the structure of DNA. The nucleic acids buddy up to form long chains, not as long as the ones found on 2 quanes. These nucleotides are made up of 3 basic components. That means there are no crazy exponents, making it simple enough to understand even if you ain't acquainted with the 5 carbon sugars, phosphate groups, and nitrogenous bases that help make up all your faces. Now what DNA does can store up quite a buzz. Not only does it store all your traits, but it also duplicates. So it can pass on those genes, which make your hair black or brown, and can determine just how much you frown. All this info must be precise, otherwise you might have to pay the price if your DNA does not replicate correctly, because that's when this is done, it can directly affect you, causing a mutation. Not the kind you get from radiation, but maybe just as severe. But have no fear, most mutations have no participation in your body and can go unrecognized. Yep, that means that they are all disguised.
Steps of meiosis, prophase 1, the splitting and crossing over of tetrads, then DNA consent occurs in the nuclear envelope and nuclei disappear and the spindles start to form. Metaphase 1, tetrads line up on the equator, the spindle has completely formed. Anaphase 1, tetrads pull apart in the chromosomes with 2 chromatids move toward the poles. Telophase 1, chromosomes with 2 chromatids deconsent and the nuclear envelope reforms around them. Prophase 2, the nuclear envelope and nuclear disappears and the spindle is forming. Metaphase 2, chromosomes with 2 chromatids line up on the equator, the spindle is fully formed. Anaphase 2, chromosomes split so that a chromosome with only 1 chromatid head towards the end of each cell. Telophase 2, chromosomes with only 1 chromatid deconsenting get surrounded by new nuclear envelopes. The 4 daughter cells are now haploid and have the right amount of DNA. They're ready to develop into sperm or eggs and become full-grown gangsters. As the chromosomes are moved into the nucleus and appear as a network of long, thin strands called chromatin. Prophase, the chromatids remain attached to one another at a region called the centromere, but each contracts compact, tightly coiled body. Metaphase, the chromosomes congregate at a plane midway between the 2 ends, which the spindle tapers. Anaphase, the 2 chromatids of each chromosome separate and move to opposite poles as if pulled along the spindle fibers by the centromeres. Telophase, new nucleus envelopes form around the 2 groups of daughter chromosomes. Cytokinesis, which may begin before or after mitosis is complete, finally separated the daughter nuclei into 2 new individual daughter cells.
Facts, you better get out of my way, telling you that genes are made of DNA. That's short for deoxyribonucleic acid. Genes are count for mutations, and that's ratchet. Genes determine organisms' characteristics or traits, but here's where stuff complicates. When consumers eat food off blades, into DNA nitrogen incorporates. DNA codes for RNA, which in turn codes for a protein, and that's where you get all your traits, which can be seen. Individuals have 2 copies of each gene, one inherited from each parent, even if they're mean. When 2 copies interact with each other, that determines an organism's characteristics. It doesn't matter whether a gene comes from a maternal or paternal side. That's proven by statistics. What matters is how many of which type of the allele the individual gets.
Replication goes down in the nucleus. The double helix is split up to strands that acts as a template for the next strand. Bases are matched to new partner strands by DNA polymers. DNA replication is the process of producing 2 identical replicas from one original DNA molecule. Transcription is the process of which DNA is copied into mRNA and nitrogen is recycled into RNA and happens in the nucleus. Translation, the mRNA is translated in polypeptide chain and the help of RNA makes up ribosomes. The tRNA nitrogen recycled as part of the amino acid. This takes place in the cytoplasm. Codons match up with the anticodons. Transfer RNA takes the amino acids to grow peptide chains and then the proteins
sweet love of the house. Just when I thought life was a breeze, I found out that I had Huntington's disease. This stupid mutation was causing me not to feel good vibrations. This mutation is an autosomal dominant, so either Ma or Pa gave me the abnormal gene. Wow, my life does not seem like a movie scene. On chromosome 4, many base pairs were repeated. I wish this disease could be deleted. Normal people have a DNA base of CAG, but I'm not normal, so CAG repeats rapidly and too many times. This disease might cause me huge medical fines. Now I have jerky, uncontrollable movements. Now I'm not sure if I'll be able to finish my home improvements. Now my moods are changing from happy to sad, now it's so swag. Now I'll be the weird kid in school. I'll sure be treated like a tool. Survival of the fittest is key for natural selection. Man, this is not heading in a good direction. I'm not an organism with variations most suited for the environment. I know I'll survive with this mutation, but the more who have it, the less offspring will be creating.